In the final video of the cellular respiration unit, we're going to do some ATP accounting in which we want to calculate what the maximum number of ATP molecules that can be produced from a single glucose molecule as our lone reactant. So to do this, we need to take a little bit of a round trip through the entire process of cellular respiration, starting with glycolysis. We first remember that ATP is not actually used as a product, but two ATP molecules need to act as our reactants to supply the activation energy that is needed to make glucose more unstable and allow it to react more readily, spontaneously breaking itself down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate here. But fortunately, after the formation of 1,3-BPGA and the phosphorus fixing, if you will, the fixing of an inorganic phosphate molecule from the cell's buffer to glyceraldehyde 3-phosphate, after the dehydrogenation here, we have created a situation where the first of our substrate level phosphorylation events can occur, because this phosphate here, originally an inorganic phosphate, is very, very unstable and ripe for ADP to come in and pick it up, producing our first ATP molecule. Remember, of course, that because dihydroxyacetone phosphate is converted into a second G3P molecule, all of these reactions subsequent to this point happen twice, which means we actually get two ATP molecules from this. And the precursor leading up to this attachment of an inorganic phosphate molecule is the dehydrogenation of this hydrogen here by NAD+, thereby producing NADH, the first of our electron carriers. But remember, since there are two G3P molecules, we also have two NADH molecules. Lastly, our second substrate level phosphorylation occurs with phosphoenol pyruvate, where the phosphate on carbon 2 of this compound is sufficiently unstable enough for ADP to swoop in, break it off of its current bond, and leave as ATP. And of course, because we have two of these molecules, because we had two G3P molecules, we also get two ATP. Therefore, even though we've produced four ATP molecules, because of the two that are originally invested, we get two ATP as our net profit, with two NADH molecules produced at this stage here. Now, we can't forget about the dehydrogenation of pyruvic acid before it joins with coenzyme A and enters the mitochondrion. During this stage, we have the dehydrogenation of pyruvic acid along with the first of our decarboxylation events, and because we have two pyruvic acid molecules from the conclusion of glycolysis, that means that we don't just make a single NADH molecule, we actually make two. Now, once we enter Krebs cycle, we can take note of all of the locations where an NADH molecule is produced. The first of one is here between isocitrate and alpha-ketoglutarate, and the second one occurs here as a consequence of alpha-ketoglutarate breaking into succinyl-CoA here, and the final stage occurs between malate and the regeneration of oxaloacetate here, meaning that per cycle there are 3 instances where NADH is produced. But remember that we have 2 acetyl coenzyme A's because we had 2 pyruvates per glucose molecule, and therefore each of these occurs twice, resulting in 6 NADH molecules per glucose being formed. But of course, we need to remember the counterpart, FADH2, that is produced at this conversion between succinate and fumarate here. So because it's 1 per cycle and 2 cycles per glucose molecule, we would end up with 2. Finally, we also need to consider this transitory phase between succinyl-CoA and succinate, in which the coenzyme APE component is broken off by an inorganic phosphorus molecule like this, which can then be claimed by ADP as another substrate-level phosphorylation, and so we have 1 ATP per Krebs cycle, meaning 2 per glucose molecule. So if we do our sum total, we can see that we produced a total of 4 ATP due to substrate-level phosphorylation, and we will put a pin in that because that seems like a gross underestimate. What we need to keep track of is the amount of ATP that is produced in the electron transport chain. So if we take a total inventory of NADH, we have 2 from glycolysis, 2 from pyruvic acid dehydrogenation, and 6 from the Krebs cycle, for a total of 10 with 2 molecules of FADH2. Which brings us to our rationalization of how exactly do we measure the amount of ATP that is produced per NADH molecule. So firstly, let's remember that we have 10 NADH molecules per glucose and 2 FADH molecules per glucose here, and this is our starting point. Now remember that NADH is able to react with the enzymes in complex 1, because this is, after all, NADH dehydrogenase, which means that the electrons that are released by NADH will stimulate 1, 2, 3 of the transport proteins within the electron transport chain, and we say that every time electrons pass through, enough H plus is transported by each of the H plus transport proteins in order to produce 1 ATP. So because of this, because of the fact that NADH's electrons will stimulate the first, second, and third of the H plus transport proteins, each NADH moves enough H plus in order to produce 1 ATP per transport protein. So therefore, per NADH molecule, we say that each of these is able to transport enough H plus to generate 3 ATP, meaning that the 10 NADHs result in 30 ATP being produced. However, with FADH2, this is not true because its active site, or the active site of this particular protein, is the one that corresponds to FADH2 because its structure is not able to fit into the active site of NADH dehydrogenase, and we can see that the 2 electrons transported by FADH2 do not stimulate the first H plus transport protein and only stimulate the second 2. So because of this, that means that because we only stimulate these 2 H plus pumps, that means that each FADH2 molecule is said to transport enough H plus to produce 2 ATP for every 1 molecule, resulting in an additional 4. So if we add up the totals, we have 30 ATP produced from the NADH molecules plus 4 ATP from FADH2, leaving us with the 4 ATP that we produced through substrate level phosphorylation, for a total of 38 ATP molecules per glucose molecule. This is, however, considered to be the maximum amount of ATP that is possible to produce per glucose molecule, assuming ideal conditions. And as a thought experiment, consider possibilities that would decrease this number. For example, consider what cellular processes that are involved in glycolysis, Krebs cycle, and the electron transport chain that would consume ATP and result in this number being smaller. This concludes our series, not just on the electron transport chain, but on cellular respiration in general. To test your knowledge, you can take a look at the case study here involving a bacterium that is actually able to do all three stages of cellular respiration, followed by the practice problems that will test your knowledge of all three levels of cellular respiration.